Yes, okay, yeah. So I realized on the GitHub it says like dimensionality reduction. And actually, in a sense, everything we're going to do is dimensionality reduction, but it's maybe not quite what you immediately think about, like PCA or ICA or UMAP or whatever. It's more in the direction of parametrizing your data, fitting a model, looking at parameters, summary statistics on the data, which are all, in a sense, reducing your data to particular meaningful parameters, but in a little bit of a different way. So yeah, and I'm still at G5 in Leo Pachter's lab here at Caltech, so we do a lot of single-cell genomics, a lot of computational biology stuff, so that's like the theme of what I'm going to talk about here. Yeah, so, and I think some of, like a lot of these topics are very fundamental to just sort of data analysis in general, so I think they should be hopefully useful for the courses and stuff coming up too, like the modeling and causal modeling stuff, and maybe also just generally for your data analysis. Yeah. Yeah, okay, so for this introduction, basically what I'm going to be going through is like single-cell genomic analysis and why it's sort of a good basis for looking at challenges in computational biology and how you think about representing biological data, which might have really interesting features, which you may or may not want to pull out and analyze. And there are many steps in this process, which you can sort of see here, and that just starts with literally trying to collect these cells, then figuring out how to barcode cells and the molecules inside them, and then putting that through a sequencing machine and getting out something, a gene expression matrix, and then doing some analysis on that matrix to get sort of whatever the biologically relevant analysis is you want to do. Yeah, and so I think what we're going to be doing is going through the steps of this pipeline and thinking about how to use sort of mathematical models and simple statistics and that sort of thing to characterize the systems and design the systems and develop hypotheses. And I think it's particularly important because what has been a big trend in single-cell genomics recently has been to make pictures like the UMAP I showed there, where you sort of take all of your data and you make one sort of 2D or small latent representation, embedding, there's a lot of different words for them, and then try to read like many different features from this one space. This could be like continuous properties of your data, discrete properties, how the cells are organized. And it's a very all-in-one approach, which can actually in the end be sort of counterproductive and limiting. So what we're going to do today is a little bit more the opposite, so sort of thinking about features that are interesting or unique about these processes and the pipeline, and then modeling those and looking at parameters and what they tell us about the data. Yeah. So for the intro lecture, I will largely be talking about the processes that happen in sort of the more upstream pipeline, so like how we get the cells and how we get the information out of the cells. Just to give a general sort of intro into the models and stats and stuff that I'm going to be talking about. And then for the methods lecture and for the exercises and all that stuff you'll be working on, we'll deal more with the gene expression matrix, which is more the actual sort of biological data that you would do your analysis from, and think about properties of that data type. Cool. OK. So why single-cell RNA sequencing? And this comes from the tradition of bulk RNA sequencing for people who are familiar. And if you're not, the idea is that you would take a bunch of cells and get the RNA or mRNA out of them, and basically get a sort of gene vector across that whole sample of just how much of each gene is present in that sample. And actually, a lot of the methods for single-cell genomics are also based on bulk RNA sequencing methods, which may or may not actually be the most apt, given that this is actually a different type of data. So the single-cell part is important because in bulk you have, like some sample, and maybe you see some trend. Some gene B's expression is correlated with gene A to some extent. And here, I guess you see a negative correlation. But if you actually did single cell, i.e. you were able to sort of see different populations of cells that were in that sample previously aggregated, you would see two different trends. You see a completely different trend. So the actual correlation is reversed in this case. And this is known as Robinson's paradox, but there are many different names for this type of phenomena where basically averaging confounds or leads to a different interpretation of the trends in the data. And so this was an example we had from the 1964 Civil Rights Act, and it just shows the sort of importance of being able to look at the components of your system rather than sort of an average. And the point is, in the House and the Senate, if you break them down by the political parties and how they voted, you would think one voted more in favor than the other, but if you looked at the components within each of these groups, you would see a different trend. You would see yeah, I mean, in the North, basically, if you separate it by political party, one group having a higher rate of voting for than against versus the average trend, which would suggest the opposite, basically. And so to put it graphically, and this is something I'll try to do a little bit throughout the lecture, is sort of to give slightly different interpretations of looking at common analyses and that sort of thing, such as to look at something geometrically. What this is sort of like is comparing the slopes of those two thick vectors in the middle, which, one is higher than the other. But if you were actually to look at the components which sum to that vector, you would get different slopes and different comparisons depending on which pairs you choose. And for us in our lab, this ends up being really important because one of the things we're interested in is alternative splicing of genes. And so you have some gene X, and that DNA can be transcribed to mRNA and spliced in different ways. So you actually get out two different products. And so even though it's the same gene name, there's actually different isoforms of that gene. And so similarly, if you look at the components, if you look at the slopes of these isoforms, or the expression of these isoforms, they may be different than when you look at something at the gene level, where you're basically aggregating that information. And that can be important. I know, for example, since we're in like a Chen Institute Neuroscience, alternative splicing is really important for forming connections between neurons and axonic connections and that sort of thing. So you want to know which cells are expressing one versus the other, and sort of aggregating that would lose that information. Yeah, and so this is like a classical single cell metaphor, which is the smoothie analogy, and it's that basically what we're trying to do is go from bulk, which is like this smoothie which has everything in it but blended up, to something like single cell where you can at least see what the components are. And then there's also spatial RNA sequencing, which I won't talk about as much, but is very cool. And the idea is then you can also see how those components are organized in a tissue or something like that. Yeah. So yeah, okay, this is more to get across the point that there are many different ways in which you can get this sort of averaging paradox. And a lot of these are examples, I think, actually from the social sciences. But yeah, and so the idea is that this is something that is important to pay attention to, and that the different ways of grouping your data can give you different trends, which may or may not be relevant to the analysis you want to do. So with that said, the point of single cell RNA sequencing is, and you can sort of see this with it looks like the Pink Floyd symbol, but is to be able to take something, decompose a tissue, an organ, a whole jellyfish, whatever it is, and identify the different constituent parts. So the different cell types is a very common word, different groups of cells that have different properties. And then also to just figure out, in terms of molecular biology, what identifies these different groups, what are gene markers, that sort of thing. Another thing that people are very interested in is more continuous relationships between cells. So how can you infer trajectories between cells and how they progress along in their development, whatever that pathway might be. And another thing is also how can you use this heterogeneity to find sort of biomarkers or predictors for states, like disease states, that sort of thing. So relating the expression you're getting out of these cells to some phenotype of interest. So that's a lot of things. And because of that, because of the sort of many components that we're possibly looking at from a single cell data set, it ends up being sort of a good demonstration of a lot of common themes in computational biology. So one is that these are really large data sets, often, especially now as we sort of grow, data sets are growing in size. So you need algorithms which are efficient and that scale to many genes, many cells, all of these things. And then there are challenges with the statistics and the sort of models you want to use based on the features of the data, what you want to learn about the biology, what's actually happening in this process. And in general, it's a really plentiful data type. It's something that a lot of labs are doing. It's only sort of growing in numbers, and I'll talk a little bit about that also. So it's a good example of sort of techniques in big data biology analysis, I guess. Okay, so I kept saying like single-cell RNA-seq and gene expression matrix, and I want to make a couple points about what I actually mean about those. So single-cell RNA-seq is like maybe a little bit misleading. One, because we're actually sort of measuring a lot of proxies for RNA. So your cell has mRNA, but what we actually do is try to capture them, convert that to cDNA, do a bunch of PCRs, correct for that, and then we get out some readout that we are sort of assuming is a proxy for the mRNA. And the other thing is that actually a lot of our analysis often isn't single cell. It's sort of using the cells as a group to do some sort of aggregated analysis, something like this, and that's something we'll get into with the exercises. Yeah. And then for the gene expression matrix, where you sort of have counts of these genes in each cell, it also is a, I guess, both loaded but also vague term, where expression is sort of what I talked about, these sort of counts of cDNA molecules also corrected for PCR biases, which are also sort of acting as a proxy for counting mRNA activity, like how active it is in the cell, protein production, which of course is not a one-to-one. You may have RNA, you may have no protein product from that mRNA. And then the matrix is something that we're going to come back to for the methods lecture, because in math, I guess, a matrix very generally just means sort of an array of numbers that are sort of a map between two spaces. And it's a little bit sometimes difficult to think about biological data in that sense, or how to think about it. And so that's what we'll come back to in the methods lecture, how to sort of use and think about your data as this matrix and properties of a matrix. Yeah.
Not to derail into the data, but I had no idea a gene had no precise definition.
Yes, okay, so I'm a little bit, well, yeah, no, I guess that's sort of true, because I guess it's like, yeah, I guess there's like particular sequence motifs and features and things like that that you look for. Yeah, but I guess I don't actually know of like what you would say specifically in terms of like its output would define a gene. I mean, actually, for people who do more molecular biology work, what would you say is your definition of a gene, if anybody wants to give? Okay, maybe not. I was going to open it up too. Yeah. I would say I actually think probably Lior, my PI, would have more interesting takes on this. But I think basically the term has been evolving since the idea of what a gene unit is and the Human Genome Project and what we know now about what these things do and that sort of thing. But yeah. But I would be interested to hear actually what the, yeah.
And so you were talking about the proxy thing.
Yeah.
What would be the most advanced sequencing technology for single cells? The capture rate of it? Like how much can you approximate the reality?
Oh, yeah, I guess like how well does it actually approximate, like if you did something like FISH or something in the cell where you're actually trying to get like single molecule counts. I don't have an exact answer, but I do have, so we'll get to a couple technologies that I'll talk about in a few slides. One of them, for example, is SmartSeq, where you get a lot more coverage of the reads in the cell just because of the way that they're capturing the RNA in the cell. So in that sense, there are technologies which just have much less dropout, basically, of that sort of information in the cell. So there are certain technologies like that which would be better, especially if you wanted to get better information on like isoform resolution or something like that. So yeah, there are certain technologies like that which would be better. But how exactly it correlates to like a fluorescence readout if you actually try to tag each of these molecules, yeah, I don't know how much I could say about that. But I think there are particular technologies which are better at capturing more of the RNA content in a cell, for example. And we'll get to a couple of them. But a lot of the popular ones are actually not that. So like 10x genomics, for example, actually has, I don't know if it's like, is it like 3% or something of the RNA content in the cell is actually captured. The other problem is a lot of methods are, and I will also talk about this, but poly-A capture based, so they only capture mRNA that have a poly-A tail on the end, which is usually a sign of mature. So you actually miss a ton of mRNA that's actually being produced and that's in the cell, but that is just not going to be picked up. So probably, actually, we also have, is it the next one? Yeah, so like, these are sort of the ideal things you would want in a single cell experiment, and there are different technologies that touch upon each of these. So there are actually now some single cell technologies that actually do like molecular, like fluorescent readouts as the molecules are being transcribed, and they're much better for if you actually want to be able to see how a molecule is being processed over time in a cell versus trying to estimate that after doing a bulk single cell experiment. Yeah, and then there are sequencing technologies that don't use poly-A capture, that instead use random primers. This is a better technique if you want to capture more of the RNA in the cell. There are also a lot of technologies that do lower numbers of cells but much deeper sequencing, and this is the SmartSeq that I was talking about, or much deeper like read capture. So yeah, but a lot of the common technologies like 10x and a lot of the droplet-based technologies tend to be better for getting a larger array of cells but at a smaller depth. So if you wanted to assess heterogeneity and many different populations, possibly rare populations, they would be better. But then you will miss out on the particular transcripts you might want to pick up. Like they tend to be worse at picking up transcription factors and genes like this, which may or may not be being expressed at a high level. So yeah, and so ideally, you would want a single cell method to be able to capture all your cells and be able to match single molecule resolution counts, that sort of thing. Capture every cell that you put into the assay, which is also, I think 10x has like a 50% rate, so you lose a lot of your cells in that process. You would want the whole length of the transcript sequence, which is again a thing you actually don't get a lot of times with these technologies. Yeah, and we'll sort of come back to these properties when we actually analyze these systems. But what people are trying to do now is optimize these different points, try to get closer to this sort of ideal technology. And so spatial RNA-seq is also probably, again, to the question about sort of direct molecule counts versus sort of a proxy for it. There's a lot of development in the spatial single-cell RNA-seq area to get more cells and more molecules per imaging of the cells. And so, again, we're not going to go into spatial single-cell RNA-seq, but there are quite a few people in my lab also who work on this type of data and basically people trying to improve the resolution of these technologies. And so this would be basically a more direct way to actually get at the number of molecules of some RNA being produced in a cell in its context without it dissociating the system. Yeah, and in general, so RNA-seq has just been like exponentially growing for the past, I guess that's only like 7 years. Oh, well, no, we're 2023, so like a decade, with a lot of different platforms being developed. But you can also see in the blue, there are particular companies that just dominate this trend, and one of them is Chromium. So this is the 10x Chromium that we see here. Yeah, which is both good and bad, has its benefits. Yeah, okay. And yeah, so with this production of more and more RNA-seq technologies and papers using them to generate data sets, we also have sort of a matching increase in the number of methods being produced to do analysis, which is great to see, but also can be really hard to wade through and figure out what is actually worth using, what is not. Yes, okay. So, yeah, and because we have a lot of methods out there, it doesn't necessarily mean that we now have some, we immediately have some greater biological understanding of what is happening under the surface of these counts that we're getting. And this is just an example which motivates actually a lot of the work that we do in our lab, which is that, so in the top row of pictures, these are like different data sets, but in the top row, we basically have a spike in data set. So this is like a control where you sort of know the amount of RNA you're putting into your system, your droplets, whatever's trying to capture cells. And basically, on the x-axis, you have the mean gene, or I guess, yeah, the mean expression of that RNA and its variance across the droplets. And when you look at real data sets, you know, cells taken from culture or from a sample, the relationship between the mean and the variance is actually quite different. You end up getting a variance that's quite a bit higher than the mean. And there are many possible reasons for this. This could be technical. It could be because of how we're picking up the cells. It could be because of how cells express RNA that actually lead it to looking like this. And so this leads to basically what is a big question in the field, which is how to figure out from the counts that you've got what is coming from technical noise, what's coming from biological noise and or biological processes, and how much basically can we relate what we see observationally from the counts and how they're distributed to what we know about the biological system and what we can model about the molecule counts, for example, and how well those actually work together. And so this is something that's basically a big direction in our lab right now, thinking about how to sort of look at observed trends and think about how to explain them with biological models, which we won't talk about as much today, but that's sort of the main question that I think our lab is dealing with. Yeah, okay, so, oh, I paused it, so I don't know how much time, but. Yeah, okay, so now I'm going to go to the technical sampling part of this process and how we would use very simple mathematical models to think about what's going on here, how we would optimize the system, how we would relate it to sort of the efficiency of capture and cell capture and mRNA capture and all of these different things. And the concepts that I talk about here will also be very useful for when we get to the gene expression matrix analysis. Yeah, okay, so yeah, there are a bunch of many different technologies that exist, and there are different reasons for using all of them depending on what you actually want to get out about your system. I'm not going to go into the details about all of these. I will just touch on a couple. And so one thing that is obviously very important to any single cell method is getting the cells in some way where they are separated and individual. And there are different ways of doing this. And so SmartSeq, which was the thing that I was mentioning, the way that it works is that usually using FACS or something like this, you sort the cells into individual wells on like your PCR plate, and then you make an RNA library in each well, and then you capture, or you basically make a cDNA library from those transcripts. And so that's what's nice about that, is that it's over the full length of the transcript, so you get, you know, the whole sequence. It does use poly-A priming, so unfortunately largely more for mature mRNAs, but that's a detail that is maybe only interesting to some people. Yeah, and so you get you get an order of magnitude better sort of capture of these RNAs than like a lot of other single-cell technologies. And this is just the details of how they actually make the library in each well, but I won't talk about those this in detail. But what is nice is that you can do very small numbers of cells, which is not the case with a lot of these big high-throughput sequencing methods. The problem is you can't actually do a ton of cells, and this is something that is being developed and being improved. But for example, there are a lot of technologies where people are assaying millions of cells at this point, and that's not something that's currently possible with this technology. Yes, but you do get the whole sequence of whatever this mRNA transcript was, because you're doing sort of one cell per well, you just don't have this sort of problem of like doublets and things like that, unless you have very, very sticky cells that you can't dissociate, but that is another problem. So what has happened is that there's a development of a lot of droplet-based technologies now, and what that means is that they create, I think this is a hydrogel bead example, but like little beads. And what they do is sort of flow these beads through a microfluidic device and push them out such that they form and encapsulate the cells, which are also being run through this microfluidic device, and a bead. And yes, and so what is in this little droplet is a lysis mixture. This will break open your cells and release its mRNA content. That in itself can also lead to loss/degradation of mRNA, but we won't talk about that. And on these beads, you have these little oligos. And what a lot of times they have is a poly-T to bind to the poly-A that's on the mRNA, or at least on the processed mRNA. And a cell barcode, which is this, which tells me I was in this droplet. So you can relate the cell to all of its mRNA, basically. Yeah, so this droplet basically creates a little reaction environment so that you can capture all the mRNA in the cell. And then this is, again, how they make the library from this for whoever is interested. Yeah, but I think the main points here is that you sort of capture the mRNA by a poly-A tail. You need to have a cell and a bead to basically be able to read out any of this information from your sequencing data. And so what this sets up then is you want the system, this microfluidic device, to do this. You want to have one bead captured in a droplet with one cell. If there's no cell, it is fine. You just, you didn't capture anything, you won't sequence anything. But what you don't want is two cells to be captured in one droplet, and now you think their mRNA all comes from the same one cell, even though they're different cells. And these are just other examples of things you wouldn't want, because it would make it harder to read out what is mRNA content from one cell in your sequencing data. So what we're going to do a couple of examples of is how to model certain properties of the system that you would want or not want, using sort of very basic statistics models. So one of the things you would want to possibly model if you're designing the system, 10x, for example, has thought about this a lot, and they already have this designed very well. But let's say you're working with a technology that's more open source, or you're designing this yourself. You would want to think about the number of barcode collisions that are going to happen. And what I mean by that is that these blue beads, which shouldn't have the same color, but have these oligos on them, and they have this cell barcode that I talked about, which is some length, and it's made of A, T, G's, and C's. And so depending on the length of that barcode, you get some 4 to the length possibilities of cell barcodes you could have. And so that means if you put even more cells in, you're going to end up with some that have the same barcode. That's bad. You don't want that. So you can try to design or look at your system and decide how often you're going to get clashes like that. If that's too much, if you can't really correct for that sort of error in your system. And yeah, OK. So what that means in terms of what you would want to estimate is, like, given some m barcodes that you have, like 4 to the length L barcodes, how many cells sort of on average would I get that are uniquely barcoded? And so we're going to formalize this using the expected value and random variables. And the expected value, let's sort of think about it as what I just said, the sort of weighted average that I would expect, is associated with the random variable, which is also sort of a little bit of a confusing name. And I think maybe it's easier to think about this random variable as like a function. And what it does is it takes your space of possibilities. So a barcode could be absent or present or absent in a droplet. And your random variable x maps that to 1 or 0. So it maps the set of possibilities to a numerical value. And so this can represent any events that you're interested in. Here we're just talking about a binary. It's present or it's not present. Yeah, OK. So what we're going to prove is that the sort of expected average number of cells with unique barcodes is going to be this number, given the number of cells n and given the number of barcodes m. And what we can start with is, OK, so for one barcode, what is the probability that it is associated with k cells, but not the other n minus k by proxy? And so what that looks like is this is the binomial distribution, which I'm sure some of you have seen before. What that means is for this barcode, there's n choose k possibilities. So there's some sets of k cells that it could associate with some probability. And then also for the n minus k other ones, it doesn't associate. And so this is the probability of, for a barcode, it associating with k cells. So what we're actually interested in is how likely is it to just associate with 1, right? Because I want it to only barcode 1 unique cell. And so what that would mean is essentially just setting the k to 1 here. So it only associates with 1, and then all the rest are not associated with it. And that gives us this formula. So then we can go back to what I was talking about with the expectation. And what I mean by it is like a weighted average, at least if you have discrete outcomes, is you have some random variable x, and this is the presence or absence thing we're talking about, maybe whatever you want it to be. It can have a value x1, it can have a value x2, up to x, whatever it is, k. And each of those have some probability of happening, p1, p2, whatever. And so this is how you get the sort of expected weighted average of that variable, whatever it represents. So let's say we have x, or xj, is representing for some barcode. It is 1 if that barcode is only associated with 1 cell, and it is 0 otherwise, which could be many scenarios, but that we don't want them. So they're lumped together. And so what this would look like then is that x could be, maybe I should, OK. So this could be 1, right? And then the probability of that is what we just calculated on the previous slide. Or it could be 0. And I guess it doesn't matter what the p2 is because this is 0 times something. But this would be 1 minus p1 because we only have two outcomes. And so that's why it ends up looking like this, because this is just 1 times the p from the previous slide plus 0. So there's nothing else. And this is for one barcode. So what is also nice is the expectation has a nice property called the linearity of expectation, which is that for the sum of random variables, the expectation can be basically broken down like this into independent sums. And this works for continuous distributions as well. You can sum the integrals separately, for people who are more familiar. Yeah, and so what the thing is now you have m barcodes, so you want to see if it's uniquely barcoded for all m of them, so you can sum them together and basically do their expectations independently, which ends up just being m times the previous slide's expectation, because there are m barcodes. So each of their expectations are going to be the same distribution. And so then in the end, you get what I had said on the first slide, which is this formula. And so what we have done basically is that since we have the expected value for all of the barcodes being uniquely mapped, this gives us the expected number of cells that are uniquely barcoded with that set of m barcodes, if that makes sense. And so that gives us this function, yeah.
Why do we assume the prior to be 1 over m?
Okay, so that is just for the sake of this example. You're just assuming that basically any barcode is equally as likely to bind. So for some so P is that probability barcode is associated with cell. We're just assuming any barcode is equally likely. It could be that it's actually sequence dependent, and you might actually want to put that into the model and think about that, or determine if this model doesn't work. But for the simplicity, it's 1 over m for us. Yeah. And yeah, I think actually generally that tends to work pretty well, especially for systems like big companies like 10x and stuff like that, who spend a lot of time optimizing these things and trying to make sure that you don't have a lot of bias introduced in these steps. There's also generally a huge surplus of available barcodes to the number of cells that you're inputting to make sure that there's basically a big buffer, which is why you can't or should not overload cells into a 10x device. Yeah, and so then what this would give you is this collision rate that I was talking about. So you have the expected number that would have unique over the total number of cells, and so 1 minus that would give you the collision rate, basically, how often you would have a problem, not what you want, not this unique barcoding. And so what we can do is basically apply those same concepts to the droplets. And I think this is basically the last main example we will talk about. But the idea is, so here we were talking about barcode 1-to-1 with a cell. We want this matching. But also, when we create a droplet, we want 1 cell and 1 bead to be in there. So there's a similar sort of process. We want a 1-to-1 matching of those. And these are examples of different properties of the system we might want to optimize or think about, usually things we don't want and want to have low probability of. And so here we have some n droplets. And so we want the probability. So you have n droplets. What's the probability that k cells are captured singly in a single manner by these droplets? And so P here now is like probability of a droplet capturing a single cell. And so there's, out of these, n droplets, the k cells could go in, some combination. And we end up using sort of the same model, the same binomial model, which models this capture of the cells to the droplets. And what is nice about this is that this distribution actually, when you're in particular regimes, and this happens with a lot of different distributions, approaches another distribution, which is the Poisson. And this is a very popular distribution in single-cell genomics, particularly also because of its interpretability. And what I mean by that is if you say x is a Poisson random variable, sort of usually what it is referring to is that x is sort of the number of events in some time period. And this could be like number of thunderstorms in a week. And your lambda in the Poisson distribution, which is your only variable, your only parameter, would be like the sort of average number of thunderstorms in a week. And let's say it's 3, and we live in Seattle. So then Yeah, basically you're modeling the average number, you're modeling the number of thunderstorms in a week given some average rate of that happening in some time period. And what is nice for this distribution is that actually the expected value is the same as lambda. So its mean essentially is lambda. And what we can do is then interpret the droplet bead / cell loading as this distribution. So in the event, basically the events are loading of a cell in sort of your encapsulation time. And you want, for both of these cases, the lambda or the mu to be 1, because you want in this event or in this loading there to only be one event, only one capture for both of these. And yeah, so this I'm not going to talk too much in depth, but basically what happens is if you use the Poisson formulation, you get nice formulations for these different rates that you're interested in. And these are sort of the things that people sort of look at when they're designing these technologies, which we actually have someone in our lab who is developing sort of a more open source framework for doing this droplet-based capture for single cell platforms. And yeah, so there's a lot of work that goes into optimizing this type of system so that you lose less cells or you have less doublets. Everything is sort of a trade-off. The materials of the beads themselves end up being different in a lot of these, although I guess we don't really know what 10x is doing. But yeah, and so this is an example where you sort of see a test of the droplets being formed in I think just capturing beads in this example. And what's nice is some of these, I think the InDrops platform actually have what's called sub-Poisson loading, where in a Poisson distribution, the mean is equal to the variance. So they sort of move together. But in some of these systems, they actually have the variance less than the mean, which is nice, because then if your mean is like 1, you're more likely to get 1 every time, rather than 2 or something that's higher or different than what you want. Yeah, okay. And so the other thing is you can also model the rate of getting technical doublets, having 2 cells, having 2 events, essentially, at each capture time. And this is why, for example, in 10x, they have guidelines for how many cells you can load at one time, because otherwise you're sort of increasing that lambda. You're increasing the number of events in a particular capture time. How much time do I have? I have like 10. Okay, so yeah, I mean, so there are different ways to look for this occurring in your data. So if you have 2 samples, let's say from the fly and from, I actually don't know what the other thing is. Do people know what the second thing is? It's okay. Anyways, 2 different species. And so you can look at the transcripts basically that you capture in each of these droplets. And you can see that, for example, there are a lot of cells which have had capture of both the cells from different species, which is not what you want. It does become a bit harder to parse what are biological doublets, so where cells before the microfluidics, et cetera, are just stuck together because they happen to be very sticky. There are other methods that people are trying to develop to address this problem. A lot of it is based on having very strong markers for each type, and so trying to see if there are cells that have both, or trying to simulate what a doublet would look like from different pairs of cell types, and then seeing how well your data looks like that or is not like that. So this is a little bit more of an open problem that people deal with. Yeah, and so basically what we've seen is with the model, for each of these different situations, you have different parameters which basically define them and which you can tune to hopefully have a more ideal system for this. So the same thing applies to the unique molecular identifiers. So this is where I was talking about PCR bias a while ago. In addition to the cell barcode, you have a sequence which is you need to have enough to encode all the molecules that are released from the cell in the lysis. Because you want to be able to basically, actually maybe I don't have this here. Oh, okay, I'll go more into details. But basically it allows you to correct for PCR amplification that may happen in a more biased way or not, depending on the transcript and the sequence and all that sort of stuff. So in addition to a cell barcode, you have a tag for the mRNA molecule itself that distinguishes it from the other molecules from that cell. So you can use similar modeling principles there. There's other biases in this technology, so a lot of them do the poly A capture, usually from the 3' end of the molecule, can be 5' capture, but that's different. But what happens is with these different capture procedures, you only end up getting particular parts of the gene, or the mRNA. So if this is the whole length of the transcript, you're largely only picking up this part near the 3' end, which can be important or not important depending on what you're trying to learn about your system. Partially because a lot of diversity can be at the 5' end, for example, of your gene or something like that. This is also something we work on modeling in the lab too. So how to, in your model, account for the fact that your sequencing platform or your cell collection platform is picking up transcripts in a biased manner. Yeah, and so there's also sequencing errors to account for. This is something that's also particularly prevalent in a lot of long-read sequencing technologies, if people are interested in that. And that idea is, again, so that you can sequence more of the transcript without having to break it up and do these short-read sequencing technologies. And a lot of optimization is happening there to reduce the sequencing errors, since those technologies tend to have really high sequencing error rates, like of the base pairs themselves. So these were the technologies we talked about. And these are just droplet technologies, and even within them, there's a lot of differences and a lot of different ways in which the library is actually produced, a lot of different ways in which you can lose transcripts or bias the transcripts that you're picking up. And then this brings us back basically to what I was talking about in the beginning, where there are a lot of different things happening in these counts, even though it looks like a very simple gene, like cell by gene count matrix. And it's basically sort of an open problem for us in the field to think about which parts of this process are leading to different patterns in these count matrices. How can we say what is coming from biology, what is coming from the capture, the 3' bias, whatever it may be. Yeah, so in general, our lab is trying to meld the two. And I'll talk about the negative binomial distribution next, which is kind of an interesting one, which can both come from just observing the data, but also can come from an actual model of transcription and how mRNA is actually produced from a gene locus. And so that's kind of cool. Yeah, okay, so that's the end of the introduction lecture. And this, we're going to be doing things in Colab, which I think at this point everybody is probably very familiar with. But this was just some links we had here if anybody wanted help with that. But yeah, so that's the lecture.
Can I ask a naive question?
Yeah.
So a lot of the question,
Mm-hmm.
network, technical noise, or biological naive question. Could you not answer this to some degree by just counting slower? Like, just one, like, as to give yourself like a Rosetta Stone of here's what the expectation is if we don't use these advanced technologies. And we just do this very carefully and look at the gene expression in what we know is a single cell. Look
Yes.
at the microscope. Do this all very
Yes.
carefully. And we get like a promise. Here's what the stochasticity is.
Yes. Yes and no. So there are both those. Okay, so I will say this also is a bridge, not necessarily because we can't do it, but because there's so many methods coming out and so much data coming out, people just tend to do this. But actually, there is a lot of work in this field which actually demonstrates that this is expected because it comes from this sort of system. And it allows you to actually build models from the ground up, which is what our lab works on. But I think this bridge actually comes more from a philosophical difference a little bit in the field, or sort of the approach that's being taken to model. There are some things, like for example, there was a big debate about whether we needed a zero inflated negative binomial, which means there's extra probability there's dropout. But there are a lot of data sets out there which do very more controlled experiments where they just input a known amount or concentration of RNA into each droplet. And so you can measure things like how much noise is actually being introduced by that. And there's different sort of controls for this. And actually, it turns out you don't really need to do that. You don't really need this zero inflated extra dropout term because it's not really something that is true to the system that people have investigated. So I think it's not that there don't exist ways to parse these out. It's just that sometimes certain things also catch on, and then it's harder to change how we model things from the ground up. But there are also other parts of it that are a little bit harder to model, like with the 3' bias. I'm just trying to think. Yeah, there are some things basically that are easier to design a control test for than not, but this example of the negative binomial is one where there is actually a lot of literature in the fluorescence microscopy field of this is the molecule being produced over time, like here's what it looks like. It has this type of profile. It leads to negative binomial counts, something like that. Yeah, so did that answer your question?
Yeah.
Hopefully I was answering the... Yeah, okay. So if there are any other questions, I'll take them. I think we have, there's a philosophy section next to it, and we had some, I think we had some examples of things that we could talk about, that I could talk about, people have questions on. We can also, maybe after that, start some of the exercises, the handwritten ones, at least the ones that are related to what I just talked about here. Which would also be in groups. So hopefully we can have people working together with the people that they're sitting next to. Yeah, but maybe I should pause. Is there like a break time? Okay.